What is gene editing for hair loss? Explained. Imagine a future where hair loss could be addressed at its very source, not just with topical treatments or hair transplants, but by directly modifying the genes responsible for hair growth. This is the emerging promise of gene editing technology applied to hair loss. But how does it work, and what are the scientific principles behind it? Gene editing involves altering specific sequences of DNA within cells to correct or modify genetic information. The most well-known tool for this purpose is CRISPR-Cas9, which acts like molecular scissors capable of cutting DNA at precise locations. In the context of hair loss, researchers focus on genes that influence hair follicle development, hair cycle regulation, and the production of proteins essential for hair growth. Androgenetic alopecia, the most common form of hair loss in men and women, is influenced by genetic factors that affect how hair follicles respond to hormones such as dihydrotestosterone. Scientists are exploring ways to modify these genes to reduce sensitivity to hormones or to promote the regeneration of hair follicles. For example, by editing genes that regulate the growth cycle of hair follicles, it may be possible to extend the phase where hair actively grows or to prevent follicles from shrinking over time. The process involves delivering the gene editing components into the scalp, often through viral vectors or lipid nanoparticles that carry the CRISPR system directly to targeted cells. Once inside the cells, the system locates the specific gene sequences and makes precise alterations. These modifications aim to restore normal function or counteract genetic predispositions that lead to hair loss. Current research is still in the experimental stage, primarily conducted in laboratory settings and animal models. Scientists are working to improve the safety, efficiency, and precision of gene editing techniques before considering human trials. Ethical considerations and potential off-target effects are also under careful review to ensure that such treatments are safe and effective. In summary, gene editing for hair loss involves using advanced molecular tools to directly modify genes linked to hair follicle health and growth. While it holds significant promise for future therapies, ongoing research is essential to turn this scientific innovation into a practical, widely available treatment option.